is nanotechnology applications in food processing, including nano encapsulation and bioactive compounds and also probiotics. And so far, he has guided 6 PhD and 14 MTech dairy engineering students. And also, he has published so many papers in peer-reviewed journals. His H index is more than 25 and I10 index is more than 40. So he is such an eminent scientist. We have it today. So on behalf of TNA and my own behalf, I welcome Dr. Hartwin Amaladas to hear his lecture.
Good afternoon, all of you. Yeah, respected Dean, the head of the department of food process engineering, Dr. Balakrishnan sir, and all madam, and all the other faculty members and students here. A very good afternoon to all of you. As sir mentioned, and as my senior also mentioned, I'm an alumni here. So I feel really privileged, and I feel it is an honor to be back here to stand before you because I'm a scientist because of TNA. Yeah, right. Yeah, I'm yearning today, I'm living today a decent life as a scientist in the society. It's all because of TNA. Okay. So thank you, TNAU. Yeah. Now, my difficult part of the today's lecture is that I'm going to deliver a lecture on nanotechnology in a place which is very renowned for nanotechnology. Okay. So today also in the morning, I visited the nanotechnology department because I consider our Subramaniam sir my guru in nanotechnology and a collaborator as well. So we have we are he's a guru, he's a collaborator, and he's also a rival. Okay, so in publishing, so I met him today. Today also I saw all the labs there. So many times I have been there, but today my lecture will be different from what they have done. I think deans are also told same thing. So my focus and emphasis will be on applications in food, where the nanotechnology center mostly works on applications in agriculture. So food is a small part of that. So we will, but I have oriented my lecture in such a way that I have given some experiments so that you can also plan similar experiments. That is the idea. So I have not given like in a very general lecture. I think my job was like made easier by Dean sir. He was telling this will give you an opportunity for others also to look at different perspective in nanotechnology so that they can apply some of these principles in their field of research. That was the idea. So I have organized my lecture in terms of some experiments and some findings so that the students find it comfortable. Oh, he has done some experiment like this, so we can also do something like that. Okay, so I have not come here with a general lecture. So I will touch upon maybe a few experiments which we have done at our place, and then what are the results we have got. Okay, so I think we will start okay. So with that, thank you very much once again for sparing your valuable time and be here. Okay, yeah, I think I'm comfortable. Yeah, fine. Yeah, let us start with the definition of nanotechnology. The Royal Academy of Engineering from UK defines nanotechnology as the design, characterization, production, and application of structures, devices, and systems by controlling size and shape at nanoscale. I have asked many students. Recently, also there was an interview. I asked a student, "What is the nano size?" 10⁻⁹. After that, unit, no, it is like 10⁻⁹ meter. Yeah, exactly. Okay. So we will see what we are dealing with, what size really we are dealing with, and these nanomaterials can be produced naturally or by artificial engineering. Okay, let us see that. What are we talking about? What scale? For example, a 10 centimeter cube has a surface area of 1 liter, that is 1000 centimeter cube. If you make same surface area, a 2 centimeter cube will have 24 centimeter square surface area and 8 centimeter cube volume. Okay, like 10 centimeter cube has a very low surface area compared to the volume. Okay, maybe another example I'll give. You can see here another example, a beautiful example of a carbon particle. A carbon particle of 60 micrometer is compared with a carbon particle of 60 nanometer. A carbon particle of 60 micrometer weighs 0.3 micrograms. Surface area is 0.01 mm square and the number of particles is 1. Okay. 60 micrometer, mass is 0.3 micro. That nanomaterial is a nanomaterial which has dimension at least one side less than 100 nanometers. A beautiful example is electrospun fibers. I think you know the nanotechnology center, and here also they make. Okay, so if you have a fiber, it's really long. So you don't need about the length. What is there? One dimension, width or diameter less than 100 nanometer. So that we call it as a nanomaterial. If you call it as a nanoparticle, all the three dimensions should be less than 100 nanometers. Even though nanometer starts like 1000 micron 1000 nanometers, but we don't take that as a nano. Like anything below 100 nanometer, we will talk as nano, right? Okay, so there is a difference between nanomaterial and nanoparticle. So nanoparticle is all three dimensions within that size of 100 nanometers. What changes at the nano level? What is really changed? Why? What is differing here is this surface effect and quantum effect. If you have a bulk material and you have a nanomaterial, what basically differs is the surface effect and quantum effect. Both I don't think like, will be able to understand. What is this quantum? Quantum, no, like, quantum theory. Okay. Let me explain the next slide, like what is changing. The catalytic property is changing. The electrical property is changing, the magnetic property is changing, the optical property is changing, the spherical property is changing, the biological property is changing, the biocompatibility property is changing, bioadhesive property is changing, every property is changing as compared to the bulk material. It's many, many times higher. Many, many times higher. Okay. So because every particle is changing, every property is changing compared to the bulk. So the nanomaterial is not same as the bulk material. Please remember that. That is why we will have some concerns which I will discuss in the later part. Zinc is no longer zinc in the nano form. Okay, a zinc which is essential, which is a cofactor for 300 enzymes in the body, which is very much essential. Without that, body cannot function. Could be toxic in the nanoform. Could be. I'm not telling is. Could be. Okay. So because all these properties are changing. For this only there is a beautiful example proverb in Tamil. Alavukku minjinal amirthamum nanchi. So same quantity you have, more effect here. So that is where we will discuss in the later slides, like, what is the effect of on toxicological effects and other things. But the issue here is material at nano level is very different. Iron will not be like normal iron. Okay, so anything which is essential, which is very much needed, may not be like, really good at the nano level. We have to see that. We have to evaluate it. And we'll just see a broader classification of nano materials, like, because I'll be in the future slides, putting some of those, explaining some of those terms. So we'll see. First, they call it as zero dimensional structures, zero dimensional, no dimension, because they don't carry any charge. Okay, so they have they are called as zero. For example, gold particles. They are very small, means like, they are called as zero valency particles or zero-dimensional particles. Then one-dimensional, as I said, like fibers, like so you have one dimension less than 100 nanometers. Two dimensions like tubes, nanotubes, carbon nanotubes, or like fullerene tubes, like that. And three dimensionals like nanoparticles, like so gold nanoparticles, or like some other nanoparticles like iron, zinc. We deal with iron, zinc, aluminum, like that. Okay, for different applications, not only for health applications, but for different applications. Is there any nanomaterial really in food? Yes, it is there. Okay, milk contains casein, which is in the nano form, maybe around 300, 250, 300 nanometers to about 400 nanometers. Okay, so casein is there, whey protein, something around 40 nanometers. And if you bifurcate this, beta lactoglobulins, alpha lactobin, they're all in nano size. So I mentioned like, they are different in the nano scale. They are different. They may be different, or they may be same, like, but, they may be toxic. They may not be toxic. So we have to evaluate. We have to have an in-depth study. Then only we can use it. Okay, in the for commercial scale, on a commercial scale. Others, like for example, like we have been, our forefathers have been cooking by gas, not by gas, by burning firewood, like and then every time they were exposed to smoke, which contains lot of nanoparticles. They were all exposed to. So nanoparticles need not be dangerous. So that is the point. Like, okay, nowadays, like we use a lot of automobiles, like cars, like so which gives a lot of emissions, which contains nanoparticles. Okay, so nanoparticle, we are living with it. In this room, there are nanoparticles. We are living with it. It's an essential part of our life. Okay, probably no politics. Politics aside, we may have to go back to firewood cooking. Okay, yeah. Okay. Why there is so much interest or concern? Because I told the properties change. So totally it is a new scope or arena to work with. So enormous potential is there. Okay. Huge gap is there because we have started now. It's not a matured science. Okay. So there is a huge gap. And things are difficult. Like, there is a difficulty in detecting and removing nanoparticles. How will, and of course, like, I will be touching upon the later part, like the toxicology or toxic and cytotoxicity concepts. Again, emphasizing what is the interest, like why there is a growing interest. You can see here in 1990, hardly there are any publications. Today, like this is like some old data, 2005. Today, this peak is still like it is. What you can see is an exponential increase, like the curve, the slope. You can see like the increase. Okay, so that is the opportunity. You know, take it in the positive way. That is a lot of people are working on. Let us join that stream. Let us see whether this in your field this can be applied. Explore the possibilities. Like that's what I'm here to tell you. Oh, okay, these are some of the examples of the experiments which we have done, and these are the results. You can also try similar things. Like okay. There are two approaches, like how to produce this nanomaterials. Like okay, so one we call it as the top-down approach, that you have the bulk material, grind it, cut it, like, and then make a nanomaterial. Okay, so for example, an engineer will be easy to tell. Like you have a ball mill, not all ball mills. Like okay, you have a ball mill, you put the micro material, keep on grinding it, you will get nano. Okay, so this is, we call it as top-down. You have a bigger particle, chop it, cut it, and get a nano size. So we call it a top-down approach. So example is mechanical milling. The second is a bottom approach, yeah, which gives a lot of creativity. Because top, you cut it, you end up with that. Like so here you assemble it. So you have a broader opportunity for assembling it the way you want. Okay, so this approach is more constructive, it gives more opportunities. Like okay, for example, green synthesis, electrospinning, or like niosomes, and a lot of other technologies there, which we will be seeing some of those technologies. Like Now coming to the topic, this is like the application. Sorry, it's the significance of nanomaterials I have or nanoparticles I have given. That coming to the applications, like so, one of the major applications is the delivery of bioactive compounds. Like so mostly it is nano encapsulation. So we have worked with say a lot of bioactive compounds. One of the bioactive compounds which is very popular is curcumin, correct? We have not worked on curcumin, like my colleagues have worked on that's different. But curcumin, then can you give some other bioactive compound? Curcumin is one, which is very good for our health, right? Yeah. Huh? Pardon? Quercetin, yeah. Resveratrol, which is there in wine or grapes. Okay. Then yeah, anthocyanins, a lot of polyphenols, like so for example, let us say like resveratrol. Wine is good. Wine is a food, a good food for health. It's a food. It's not a drink. Okay, so if you drink wine, you should never die. Because it will quench all the radicals in the body. It will it has an anti-aging effect, anti-inflammatory, antioxidant. All anti is there. Okay, but after even if you drink every day you will not get like you don't live like quite a long period you are expected because that resveratrol can have all these properties but you will not be able to realize all those properties. Why? For example, like stability. The moment the moment you take resveratrol, okay, for example, grapes contains 0.1% resveratrol. In our body, the assimilation is less than 1%. So 1 kg grapes become how much? Already 1.1%. And 1%, less than 1% is assimilated in our body. So you get minuscule, like, so you don't get any effect, really. Okay, why? Because the environment in the human digestive system. Can anybody tell the pH of stomach? Yeah, huh? 2. Yeah, closer. It's about 1.2 to 2. Can you imagine the acidity? Really, like, when we start, like, when I come to know, is it? Like, okay, so that is the environment in the acidic, like in the gut. Okay, so what happens? Even whatever goodness in reservoir, it's all get destroyed there. No use, like, whatever you have a peptide, bioactive peptide, you have curcumin, everything, it gets cleaved because it's a basically hydrolytic process going on with the enzyme, sorry, with acid. So it's a hydrolytic breakdown. So you don't get the real benefit. Okay, so that's what you have problem against pH. You'll not be able to get the benefits of the actual bioactive compound. And sometimes we all in food processing, like we heat or like without heating, most of the things we don't do, like everywhere heating, so it gets destroyed. Oxidation, light, all factors are there. Environmental factors like temperature, humidity, everything is there against it. So it is there, but we don't get it. Okay, so encapsulation, particularly in the nano form, will help to get it. We'll see how. It can help in improving the taste and color because some of, for example, peptides. We deal with milk, milk, whether it is casein or whey protein. You break down, you get peptides, and most of the peptides are very, very bitter. And if you encapsulate it, avoid that bitterness. Maybe probably curcumin also, like okay. Safety, it is good for encapsulation is good, and but nano is like we have to see that like toxicological effects like bioavailability. I will emphasize that like maybe in the next slide. Yeah. Stability against pH, you can see that in the mouth the pH is like 6 to 7. This is not exact data, but like in the gut it is about 1 to 1.2 to 2. But when it goes to the intestine, it becomes 7.4 neutral or slightly alkaline pH. Okay. Yeah, this is where I was telling why the bioavailability is a problem. Okay, so you can see here on the top, intestinal and lumen, where all the nutrients are there, whatever we have eaten, it goes to the lumen, and then the mucus layer, it goes to the epithelial cells into the small intestine, where most of our nutrients get absorbed. Now, here in intestinal epithelium, there are four types of transport of nutrients. The first is transcellular through the cell directly. This is the epithelium cell through the cell directly. Next, paracellular, I will come to that later. Carrier mediated, see the blue color, the sky blue color. So through some active compound, it gets transported. Or transcytosis through some cellular contents, like it gets transported. But if you see all that, there is it gets transported, and you can see that bottom red channel, that is your blood circulation line. It enters that bloodstream and gets into the body. Now, all that processes like A, C, and D require energy. Okay. But what about this process B? This is where nanomaterials have an advantage, nanoparticles have an advantage, or nanomaterials. What do you see those green patches here? These are called tight junctions in the intestine. And these nanomaterials are small, they can open up the junction. So it is like coming into the hall, buying a ticket or free entry. Which will be? Audience will be more? That depends on the speaker, I think. Okay. Right. Or depending on the movie, right? If you go to a hall, which will be like, that is a free ticket. The nanoparticle movement takes place by paracellular route. No energy is required. It is opening just a gate and entering it, not going through a lengthy process like to reach the bloodstream. So here is the better chances of bioavailability. But I come back to that Tamil proverb. அளவுக்கு மிஞ்சினால் unlimited things can enter. So spike can happen. Okay. So, somebody was like having seizure, and the doctor recently, one of my friends son, like he said, don't give us protein spike diet. That means a diet which is very rich in protein. You have to distribute it like so. Any spike is not good. Okay. Here that risk is there because it is a freeway. It is a freeway, no gate, no parking, nothing like this thing. It's a freeway. So that paracellular route is a freeway which does not require any energy for absorption of the nutrients. So that is it's like a double-edged sword. One thing is it can increase the bioavailability, yes, but as I mentioned, the proverb, it can lead to that also, the second risky part. So you require a balance. You can see here, this is the tight junction. These are all borrowed from internet. This is a very recently published paper in ACS. Like you can see that on the, okay. So that is what I just go back here. Those green dots here, those are the junctions which I have detailed it here. Okay, and those junctions get opened up, and that much is enough for a nanoparticle to enter into the bloodstream. Whereas no other material in micro or any other form can enter without that, by opening that watertight junction. Sorry, not watertight, the tight junction. Okay. So this is where there is a tremendous opportunity if you involve like nanotechnology in a food system. Assimilation, bioavailability, everything is increases tremendously. Like, okay. So let us see the risks like later. Good things first. Second is control release. You can release in the targeted way. I was telling about the pH in the gut. Avoid it. We can possible. It's possible. You can encapsulate it. Okay. For example, we are doing something with pullulan, which is like, is a starch derivative. It can be produced by the fungus also. Okay. But it will not dissolve in water easily. It takes about 3 hours, like to dissolve. Okay, even whatever be the pH. So if I use that material for encapsulation, it's a carbohydrate strictly. It can be used for food application, no issue. But if I use that, I can bypass the stomach because I am not going to, if I encapsulate any bioactive compound using that pollulant, I can, by the time like 3 hours passes, I will be able to, because food stays in the stomach for 2 to 3 hours, I can pass the stomach. So where assimilation should happen, that is in the intestine. And where we can directly deliver your bioactive compound, whether it is like resveratrol or curcumin, or directly into the small intestine, where it gets assimilated in whole. It does not get broke down, like in the stomach. And then the other applications are like food packaging materials, development of nanocomposites, like early detection of pests, like something related to food applications, not into like agriculture. I'm not diverging into agriculture. Monitoring and control, like and then quality assurance, like so where there are a lot of scope for application of nanotechnology in quality assurance as well. Coming to the work, like which we have done like earlier in our institute, like we have used many approaches for delivery of these bioactives. Like one is the niosomes, other is the proniosomes, and third is the electrospan nanofibers. And where we have used or encapsulated most of these bioactive compounds like resveratrol, quercetin, catechins, peptides. Now we are working on probiotics. Okay, delivering probiotics by nano micro encapsulation. I discussed about this, like resveratrol and quercetin earlier itself, like because of the bulk, the gut pH, the bulk form of resveratrol gets destroyed in the acidic conditions, like so you don't get the bioavailability of resveratrol. So what we do is the first approach of encapsulation is niosomes. Like it's not very complex process. So I'm just now starting with like the experiments which we have done, like so that you can also get some glimpse and then you can apply something which I'll be very grateful if you can apply it. Okay. So the first approach is niosomes, like which is niosomes is like a capsule which has bilayers, like hydrophobic layer and hydrophilic layer. And you have any bioactive compound, whether it is hydrophobic or hydrophilic, you can bind to it. So because it has a bilayer, like hydrophobic layer and hydrophilic layer. Okay, so you can bind any bioactive compound. In fact, hydrophobic substances get absorbed better in the gut than hydrophilic. Please, stomach. You don't think like water soluble substances get absorbed, but fat soluble substances get absorbed better in the stomach because the stomach likes hydrophobicity than hydrophilicity. Okay. So we have used different surfactant stabilizers, nutraceuticals, like, and then produce this vesicles or like capsules, which are bilayer containing, as I mentioned, like, the hydrophilic and hydrophobic layer, where you can encapsulate the active ingredient inside. So the hydrophilic layer or hydrophobic layer arranges itself and the material is encapsulated inside this capsule kind of thing, like which you can see here. The active ingredient will be inside. Okay. So we could produce something like particles of 200 nanometers with the particle then like zeta potential. Zeta potential is one which gives the indication of stability. Higher the zeta potential, more is the motion between these nanoparticles, and less is the chances of settling down or aggregation. So higher zeta potential is very good. Entropy and efficiency we got for resveratrol or quercetin is something about 79%. And you can see here what happened. I have just combining resveratrol and quercetin. You can see here resveratrol on the first left, like it is insoluble in water. Converted nanoform. Okay. You can see here quercetin on the right hand side, pure quercetin, and quercetin in the niosome form. You can change the solubility. Okay. You can increase the stability. You can see here the free resveratrol on the blue line, it gets degraded with time because it doesn't like light. Whereas the encapsulated form, you can see here more or less it is retained here, even after exposure to light. Release. This is very important. Sustained release. As I mentioned, like food stays in the gut for 3 hours. After that, it stays in the intestine and colon for like 21 hours. So rest 21 hours only absorption takes place. And if your nutrient is released in the next 1 or 2 hours, it gets released, everything gets broken down in the stomach. If it gets released in a long sustained way, then it gets released in the intestine, where there is better chance of assimilation or absorption. So encapsulation helps, that is niacin helps in sustained release. The red line you can see here, like it is sustained release. No change in the antioxidant activity after resveratrol, after encapsulation. Okay. So that is because you can do that encapsulation, but there should not be any compromise on its functional properties. That is very important. Okay. There's some studies which we have done in some, macrophages, which one of our students did, like, so it's not my work. You can see here, controlled resveratrol, LPS, lipopolysaccharides, like lipopolysaccharides plus resveratrol. But when we added this niosomes, all these ROS species got, all the macrophages got destroyed. That means, like, but if you add just resveratrol alone, you have, like, say this is a control. You have, like, say 25% dispersion. Whereas you put in the niosome form, that was. So the antimicrobial or antioxidant activity, those things can be improved. So taking some clues, like we have done some work in cancer cell lines. The normal cells takes little bit of resveratrol, whereas the cancer cells takes a lot of resveratrol. Okay, they have the property. So which is an advantage, because resveratrol is a very good antioxidant, RO is quencher, is anti-inflammatory. So all that can be, like, a little bit helpful. So you can see here, the cancer cells can take free form of resveratrol in after 24 hours, whereas this is an encapsulated form. You can see a lot more resveratrol can be taken up by the cancer cells. After encapsulation, resveratrol can be taken up in a large quantity by that. And you know the properties of resveratrol. So if you can internalize the resveratrol within the cancer cell, then we can probably stop its proliferation. Okay, so that those are, like, some initial trials we have got, results we have got. This is also on lung cancer line. This is like a double staining with acridine orange, and you can see here the orange spots, like, indicates destruction of those cancers. We have also, like, done some fortification studies in milk, because we have encapsulated, non-encapsulated. We have to deliver it. How to deliver it? Like, so, for example, like, milk is a very good medium for delivery. People say calcium, you drink more milk rather than getting calcium tablets and eating it, because the delivery of calcium will be better from milk. Similarly, milk proteins and some of these bioactive compounds, we have seen that the delivery system works synergistically. So we have fortified milk, and we have seen the stability of this, like, niosomes in milk. And we found that, like, they have better stability as compared to the free form, or like the normal without fortification. So that is about niosomes. So the one approach which I have discussed on nanotechnology is the niosomes. The second approach is proniosomes. As I mentioned, like, the niosomes is a liquid form which has like stability for a month, not more than that. So if you want something longer, then all food processing people know that you have to dry it into a powder, right? Correct? Yeah, dry it. Dry form is better. So that is, we call it as proniosomes. Here, for example, we have worked with, like, catechins. You can see, like, tea contains catechins. Like, so we have used again thin film hydration. There's a technique called thin film hydration. Then we have produced, like, we using stabilizers and surfactants. We have encapsulated that catechin into that. So you have a same, lyosome, like the vesicle and the the active compound is encapsulated and we can get a dry powder. Okay. So now you want to any add in food system, you can add it and use it. At that time, you can convert it to the niosomal form, the real nanoform. This is not in real nanoform, but if you dissolve it and homogenize it, you get a nanoform. Okay, but other than that, it is a long time storage solution for that. We use different wall materials like maltodextrin, lactose, pullulan. You can see that this AFM gives the variation in the size and all, like there's not much variation. You don't see huge peaks there. And this is a TEM image. We can see that you can see those. These are the niosomes which are spherical in shape. You can see that indicates the spherical shape of the like these catechin niosomes. Like we have seen the structure. You can for example, we have done XRD. We can see that the original peaks of resveratrol is retained. That means the functionality will not be lost. But FTR said that like your groups on the surface, they're covered. That means like your active ingredient is encapsulated, but the core principles of the properties or functional properties of the active ingredient are not lost. Okay. The third technique we have used is electrospinning. I'm not going like you can see here. We have got a very beautiful image of the spray which got. Yeah, this is a thermal image. One of the main advantages of electrospinning as an encapsulation technique is that it can be done without any harmful solvents, no chemical solvents. At least we have come out with 4 or 5 food grade polymers. Food grade polymers. We have 4 or 5. Okay. And all are mostly water based. So we can use only water for electrospinning. Okay. So, and this process happens at ambient temperature. So there is no harm for any of these. Even I'll cover it later. We are able to encapsulate probiotics. Okay. Without, which is comparable to that of freeze drying. So we have electrospun, resveratrol, catechins, and peptides using, as I mentioned, like, these are some of the wall materials we have used. You can see here, all are food grade, food materials, not food grade, food materials. Like, okay. I'm not going into the advantages of encapsulation. Okay. But like, we have also like, failed with some of the things like soy protein isolate didn't work, like pullulan plus combinations. No, pectin plus pullulan, yes, works. Like, so we have explored maybe about 25, 30 combinations, like, so you can see here how beautiful, like, clean fibers we can produce, which contains the active drug, okay, or the bioactive compound. And just you can see here, this is the fiber diameter which we have got, so less than 100 nanometers. And the encapsulation efficiency, I think one of the best techniques to get the highest encapsulation efficiency is electrospinning. Okay. We also evaluated the antioxidant and anti free radical oxygen scavenging activity. This is for peptides. Okay, we encapsulated by electrospinning. We found that like there is not much deviation or much change in the antioxidant activity. We also did coaxial electrospinning. So one, I'll come to that. This is an interesting experiment. So we got something around 94% efficiency encapsulation, but we want to even go higher. So what we did is like coaxial electrospinning. Outside is purely the food polymer, one of these polymers. Outside, the inside contains the rich in like the bioactive compound, whether it is peptide or like resveratrol or catechin. Inside is like very high concentration. Outside is a free polymer. Inside also contains some amount of polymer. Like polymer means like the food grade material, whatever I have, food material, whatever I have mentioned. Like so, with this we are able to again, if you don't get the factors correct, you will have some of these like droplets there. But if you get it correct, you can see the idea is you have a very wide range of operating conditions you can achieve for encapsulation. Okay, you can see different conditions like beautiful, clean, homogeneous. You can see the variation in diameter, hardly any. Okay, so such very good, things we can, very good fibers we can produce. And this is the AFM image. Like, if you get the conditions correct, like neat, you can see the 3D network structure. Yeah, very beautiful, like, fiber like structure you have got. Yeah. And this coaxial electrospinning, I told, like there are two nozzles, like, one inside another. You may have an apprehension like whether the diameter increases, whether it's above nanoscale. No, still we are able to, like, get this same diameter. We were able to achieve that. Hardly any difference in the characteristics of the fibers. Yeah, this is the nozzle. Okay, coaxial nozzle. And yeah, and this is one experiment we did was X-ray photon spectroscopy, not photoelectron, X-ray photon spectroscopy. And here we did it for carbon when we did single and coaxial electrospinning on top. Okay, single and coaxial on top for carbon. For nitrogen, we have encapsulated peptides. Peptides contain nitrogen. So we are finding out how the two different encapsulation worked. So when we take nitrogen, because we have encapsulated peptides, so nitrogen is there in single. You can see it was like, wrongly plotted in single. In that red line, you have nitrogen peak there. So some amount of peptides are sticking out of the fibers to the surface. So that is why the encapsulation efficiency was about 94%. In coaxial, there is no nitrogen peak at all. That means we are able to achieve nearly 100% encapsulation with coaxial electrospin. Okay, so there are a lot of opportunities here to do electrospinning. And we also have seen the in vitro release. You can see here, very slow release. In the gastric stage, we have simulated the gastric conditions and also the intestinal conditions. Like, we also have fortified milk with this encapsulated peptides, and we didn't see any difference in the taste. Actually, I mentioned in the beginning that peptides are very bitter. So if you add fortified milk with peptides, you will get the bitter taste. If fortified with electrospin encapsulated peptides, no change in milk, the taste of milk. Okay, so this is one medium for delivery of bitter constituents. Yeah, I think, we got like something around 97-98% efficiency when we, like, same condition when we used. Okay, and without any compromise in the antioxidant or any of the functional properties. Like right now we are handling two projects on encapsulating probiotics by electrospinning. One is funded by DST and one is funded by NASF. Where we can see that these are the microorganisms which are encapsulated. Okay, we have been working with Rhamnosus, then Plantarum, Rotary, and one more. Okay, and you can see here compared to freeze drying. In freeze drying, the microorganisms are just embedded, not encapsulated. And freeze drying is the standard, gold standard for producing dry cultures. Okay, whereas here you can see it is encapsulated. You don't see the bacterium. You've seen hundreds of images like this. You don't see the bacterium. Here you can see those bacterium here and there. They are all just embedded. So in freeze drying, and this technique can give, like, I think, like encapsulation efficiency as high as 95%. Okay, which is like even comparable or even more than that of freeze dried cultures. Okay, and but there will be a concern, like, you say you are applying voltage. There are food grade or food materials, like we call it as lyoprotectants or cryoprotectants, which can lyoprotectants, not cryo. The cryo is for freeze drying. Lyoprotectants which can avoid this voltage impact. Okay, so those are like very charged materials. They carry the electrospinning charge rather than the probiotics. So we can see that at 18 kilovolts we have population without any sacrifice in the population. But as the voltage goes up, yes, there is an impact. So you have to find a compromise, like and we can, after encapsulation by electrospinning, we have like opened up, we have dissolved that, we have centrifuged, took the microorganisms, again fermented curd with that, we were able to make dahi, no problem. So the functional properties of the probiotics, we are evaluating other functional properties which are there. I have not presented it here, but to the best of knowledge, our results so far indicate that there's no loss in the functional properties or probiotic properties of those microorganisms. We also have applied this nanotechnology in platelet exchanger. We know that milk is invariably heated in a platelet exchanger in the dairy plant, and every 8 hours there will be stoppage because there will be fouling. Because milk proteins are the worst foulants. Heat milk, like in a vessel, you see that like you heat it even little bit more after 5 minutes. You see like after you pour out milk, a lot of your valuable protein will be sticking to that vessel. Like okay, so that is a major problem, fouling. So we have, this are the corrugated plates. So we have taken the stainless steel plate. We have uh, heat treated it. Then we have applied that coating, PTFE coating. So when we do PTFE coating, that is the Teflon coating, what happens? Teflon compromises the heat transfer efficiency. So what we did, we have included titanium dioxide into the Teflon. Okay, and then we made this plates, heat treated this plates at about 900 degrees. Okay, and then they stuck very well to the plates, which we have used for treatment. So this image gives the contact angle of water on those surfaces. You can see here, this is for SS30, 316. This is for milk and that is for water. You can see the change in the contact angle. So you are modifying the adhesion property of that plate exchanger or plates. So let us see what happens on the following. Okay, this is what the originally the SS plate, which is polished. Under microscope, it looks like on the top image on the left, top left, still it has cracks. But when you use a nanomaterial, like you can see, very thin cracks are there. Okay, so that is a difference. And that can be seen in AFM image also here. And this is a surface chemistry. We have done that EDX, which you can see stainless steel, lot of minerals are there on the surface, whereas Teflon coated, you can see titanium, carbon, oxygen, again iron, little bit of fluorine probably is there. So this SS surface is now coated with titanium containing, sorry, PTFE containing titanium dioxide nanoparticles. You can see here it can be reduced by 90%, 1/10. So every 8 hours we have to disrupt the dairy industry process, like because of fouling. Because if plates gets fouled means there is going to be a lot of pressure built up. Here there's no fouling, so you can run for long hours like the dairy plant. Okay. And what happens to overall heat transfer coefficient? Initially, because we have coated with Teflon, there is a drop in the heat transfer efficiency. But once fouling happens, the heat transfer efficiency of the normal SS plate decreases. Whereas here there is no reduction. That's what the blue line is. So after some time, you have a shoot up like this keeps going just like Duracell ad. Keep going. Like I'm not a brand ambassador of that, but just okay. And this is the fouling resistance, the buildup of deposits. This figure shows like you can see that uncoated surface is the red surface, red graph where the growth of the after some time the deposits starts like increasing exponentially. Once deposition starts it, like it's like a it encourages more and more deposits, like so it will be very faster, whereas the coated surface, the deposit in or fouling resistance we call it as like very less. Overall heat transfer, I think I have discussed that, and we also have applied this like in other fields, like we have also like synthesized these metal nanoparticles. One of the interesting methods of synthesis, like we have, I think you might have like heard about a lot of plant metabolites being used for synthesis of nanoparticles. We have used cow urine, very beautiful because it has urea, okay, which is a very good reducing agent, which has peptides. Okay, so a lot of things which can be used as a reducing agent is there for the green synthesis of nanoparticles. Has anybody tried green synthesis of nanoparticles? You can synthesize in your own lab. Doesn't require anything. Yeah. Okay. So most of these techniques, whatever, except electrospinning, you don't require very high-end technologies. Still, you can produce nanoparticles. Okay. So this is like, we have synthesized alumina doped zinc oxide nanoparticles, which we have used with therminol, which is a heating fluid. Okay. So we have been working on nanofluids for increasing the heat transfer efficiency. So to increase the heat transfer performance of heat exchangers. Okay. Because a lot of heating takes place in the dairy industry, like okay, heating and cooling. We have characterized the synthesized nanoparticles. Again, I'm telling you, it's all very basic technique, green synthesis. It doesn't require complex, so like machinery or like complex technologies or setup infrastructure for synthesis. It's not required. We have synthesized and we have characterized that. We have put it in the tubular heat exchanger to evaluate the performance of the nanofluid comparison. Okay, you can see here when we increase the concentration of nanoparticles in the fluid, we have increased the overall heat transfer coefficient. When we increase the overall heat transfer coefficient, my fellow engineers will agree that it will increase the overall energy consumption will reduce, it will increase the heat transfer efficiency. Okay, so that's what you can see here. So it helps in reducing the energy consumption in heating applications, and also because it increases the energy efficiency. So the nanoparticles which we have synthesized is something like you know 20% increase in overall heat transfer U, and about 5% no energy efficiency increases by 5.4%. That is energy consumption reduced by 5 point. That means efficiency increased by about like 22%. Yeah, there is the nanoparticles, like we have produced something about 17.40 nanometer in our lab without any complex technologies. That's what. And because I do the DLS for the students, so sometimes, a lot of times, one of the students, like I'll show the next work, like she regularly gets 10 nanometers. I will be thinking, what? This is like, we take 100 and now we are working with 10. Okay. So for DNA, it is only 1 nanometer. So we are getting particles 10 nanometer, very close to that. Okay. So sometimes, like, we feel like, oh, okay, you have to go into that size, then only you will imagine what we are really working with. Okay, so some of the other applications are like zinc oxide, and from zinc oxide we have produced zinc oxide nanoparticles, like again with the green synthesis. We have used different urine plant extracts. You can see here it is biological extracts plus metal oxide solution, that is zinc oxide salt you buy from high media, put it, plant extracts. You start the reduction reaction, then you continue for some time for the reduction reaction to complete. You get the synthesized nanoparticles. So it's not going to be very complex. Yeah, Simpleborg citratus, like we have used to produce nanoparticles. Okay. And carica, papaya extract we have used. So we have used a lot of biological extracts, urine, like that, to synthesize metallic nanoparticles, which we have used for heating applications as nanofluids, and also for, yeah, this is the characteristics of that same, also used for wastewater treatment. So the applications of nanotechnology doesn't end with food also, like the food waste also can treat with. And this type of metal nanoparticles, I think I can go back to that slide. I just skipped that. This type of nanoparticles are big ROS quenchers. They can interact with enzymes. They can destroy cells because they can induce leakage in cell walls. They can interrupt the electron transport in the cell, so they kill bacteria. Not only bacteria, they can kill. They can destroy toxic compounds in wastewater. Okay, toxic compounds in wastewater. So we have been using two columns. One is the reference column. Another reverse sand. We have removed all the organic matter in sand, and then we have coated that sand with nanoparticles. And we use that as a treatment column. And we have been comparing with the reference or standard column. Now coming to the potential risk involved. As I mentioned, there are toxic and toxicological issues there. Okay, cytotoxic issues there. Unless we thoroughly investigate it, I don't recommend anything nano is the cure for all. Whether it is agriculture or food, everything sometimes enters the food system. You may develop a, nanopesticide or fertilizer or something like that. Finally, it may enter the food chain. So ultimately, you have to do the toxicological studies. Okay. Even I told you in the beginning, there is possible health effects from accidental exposure. There are nanoparticles in this room. It can enter into, through your skin. The cosmetics which you are using nowadays started containing nanoparticles. Please note that. Yeah, they can easily get into that body through the skin. Directly enters the bloodstream. There's no barrier, no protection. Okay. So, and I told you about the paracellular pathway of absorption. So there is a potentially explosive properties possible, so I invite you to recall that Tamil proverb, so everything can go freeway. It's a freeway, okay, because of the scale. But okay, this not only the toxicology effect or probably effect. There are limitations in risk assessment because how do you measure? How do you detect a nanoparticle? There is no detector. Okay, can you filter it? No filter. So how do you clean it? So very difficult to detect. For example, you put it in a particle size analyzer and see whether it is in nano size or not. You should have a nanoparticle analyzer, which costs like 25 lakhs. Okay, so it is, it requires a sophisticated equipment and difficult to predict. Till today, nobody is clear. If anybody says nanoparticles are safe, don't believe. This is what my knowledge is. Still, we have not analyzed fully the behavior in the different environment, in the gut or in the ambience, and through the skin, through this, we have not assessed it. Okay. Potential to absorb toxic. They can, I told you, because they're in nano form, the atoms are exposed, they can bind with anything. Okay. So they can bind with toxic compounds for example, you're in the soil, and it can enter the food stream. Okay, it can enter into the plant, and then it can enter the fruit or the grains or whatever it is, and then it will come back to us. Okay, so they have the potential to absorb toxic chemicals, okay, and so long-term study is longevity of these particles are required. Long-term study is required before we start applying it on a commercial scale. Okay. With this all, I'll finish my technical content, and I take this opportunity to acknowledge the funding contribution by NSF, DST, and the support they provided by the director, IC, NDRI, and my team, which consists of faculty, RA, SRF, students. And because of their work, I'm here. Okay. Thank you so much. Thank you, TNAU. Thank you once again for providing this opportunity to be here in front of you to spend this past one and a half hours with you. Thank you very much. If you have any questions, quick questions, I think we can.
I have a question for you.
Yes, sir.
Excellent and exhaustive presentation.
Thank you,
My
sir.
question is, spray drying is known for years. Amul invented 75 years ago, and we've been using. We don't call it as a nano. How exactly
Yes.
nano spray drying at this time is different from what we already knew?
How is nano spray drying different from the
No,
normal
no.
spray drying?
Yeah, sure.
Am I right, sir?
Yes, exactly. No.
Yeah,
Example is Amul milk powder.
exactly.
Milk
Sir,
powder. Amul.
milk
Amul
powder.
is milk powder. It's already
But,
known.
sir, in milk powder, I don't think anybody is working with the nanoscale. Okay, sir. Probably they may be drying some of the nutrients which they are incorporating into milk powder, maybe by nano. Otherwise, you have a nano spray dryer, the output is in a few grams per hour. Nobody is going to use it unless you are specifically encapsulating some of the active ingredients by nano spray dryer and including that into the commercial milk powder, which is produced by the normal spray dryer.
Harvin, my question is more of just for students' sake. I am asking this
Yes, sir.
question. I suppose nano has a unique advantage of surface area, as you nicely explained, and bioavailability is much better than regular spray drying or macro scale. And
Yes, sir.
consider that amul, which is already known as a spray dried material, which is being used as a food, baby food, for a long time. Suppose you make the same thing with the nano scale,
Yes, sir.
how it is going to help? Can we reduce the quantity or increase the bioavailability? How it is going to help?
No, my concern is more than bioavailability. My concern, immediate concern, will be toxicological aspects. Have we
Okay.
done at least 5 years study on it before we recommend anything? Because please, sir, history states that most of these industries have suppressed information for not years, for decades. And now they are like, they are reaping the rewards in the bad way for that. Some have to withdraw, some have to pay penalty, all that. But so I don't recommend unless there is an exhaustive study on toxicological things first rather than bioavailability, sir.
Okay. So within the nano scale products, of course, there are nano fertilizer come into the market now. But
Yes, sir.
nano food system is the major one, as you rightly indicated. Nano food system, there are 500 plus products already there in the market. Nano
Yes, sir.
packaging, nano food, Nestle has come up with the nano product. Several
Yes,
nano
sir.
products already there in the market. Of course,
Really, sir.
regulatory
Yes, sir.
framework, we need to look into it carefully. And I think we'll take forward some of the message you indicated. Uh, Thank you very much for
Thank you,
a
sir. Thank
good
you,
talk.
sir. Thank
And
you, sir.
if there is something can be done, because I know you got a major NAS project, 4.5 crore.
Yes,
You
sir.
got that. I was a technical committee member.
Yes, sir.
Just to look forward to see some of the
Yes,
nano
yes, sir. We
products.
will do it,
Make,
sir. We will do it, sir.
make sure that you validate according to the DBT
Yes, sir.
guidelines already
Sure, sir.
stipulated.
Sure,
Okay.
sure, sir.
Thank you very
Sure,
much.
sir. Thank you, sir. Thank you, sir. Thank you for the suggestion. Thank you. No, actually, the real concern is that there are no regulations, as I mentioned in one of the slides, but there are a lot of products coming into the market. In the absence of regulations, they are like, they are free. Okay. But, who will bear the impact, which can come in the later stage, we don't know. Like,
Hello, sir. Good evening, sir. Sir, am I audible?
yes, sir.
Yeah, yes, sir. I'm Selva Prakash, sir. I'm working on extrusion technology for extruded pellets for birds food, sir. It's a pet food industry. So in this pet foods, sir, am I audible?
You're because of the disturbance here, and no, I am not able to hear you, sir.
Okay, sir.
Yes,
Okay,
sir.
sir.
One
Okay, sir.
second, sir, please.
Okay, sir. I'm Selva Prakash, sir. I'm working in R&D.
Yes, sir.
Yes, sir. In this extrusion technology, we are using extrusion technology for manufacturing a pellets, sir, which is going to be feed for exotic birds. You may know Amazon, cockatoo, conus, and all is there, no? Bird food. So you can't get that.
I am not able to comprehend the question, sir. Like, I don't know.
So,
Really.
so am I audible clear?
You are audible, sir, but there is some bit of echo also, which I am not able to understand the question. Yeah, sir, my email ID is very simple, hartwin1@gmail.com. HEARTWIN, hartwin1@gmail.com. We'll be able to share whatever information we have. Because I have worked with extrusion in my PhD.
Oh, okay, sir. Okay, sir.
Extensively.
Okay, sir. Okay, sir.
So I
I'm
work-
working on extrusion technology, sir.
Yeah, so I'm not able to, sorry, sir, I'm not able to really hear the question fully. I'm not able to hear the first part, understand the first part, but the end of the question, I'm not able to comprehend.
Okay, sir. Okay.
Yeah. Because we are sitting in a hall, there are
Oh, okay, sir. Okay,
about
sir.
100 and 200 people, and their mouths are not closed. So that's the thing.
Okay, sir. Okay, sir. I will mail you, sir.
Yes, sir. That will be, we'll be able to share, sir.
Yeah,
if
sure,
you
sir.
know.
Sure, sir.
Right, sir. Thank you, sir.
Thank you so much.
Yeah, now it is better. When he was asking the question, like,
Shall I ask, sir?
yeah, your voice is echoing. Sir, yeah, ask, sir. Now it is
Yeah,
silent.
yes, sir. Yes, sir. I'm working on extrusion technology, sir.
Yes, sir.
These extruded pellets are going to be feed the exotic birds. You may know the Amazon cockatoo, that kind of birds. So in these birds, we are in the formulation, we are adding a vitamin mix, mineral mix, and also the prebiotics and the enzymes and all we are adding, sir.
Yes.
So in extruded in pellets in final products we can't get the exact amount. So what we are adding, we can get the only 20% 20% of the bio availability, sir. So when we are testing means. So is this
20% is very high, sir.
Retention, sir. Retention.
Okay.
Yeah. Okay. After
During extrusion, sorry to interrupt. During extrusion, your retention of bioactive compounds is only 20%, if I am right.
Yes, sir. Yes, sir. Yes,
Okay.
sir. Yes, sir.
Okay.
So now we are going to launch the product. So existing product formulation, we are going to improve the retention level to
Sir, I
into
got it,
22.
sir.
Yeah.
I got it, sir. Like, see, in PhD, I have published 7-8 papers on extrusion alone.
Okay.
And
Okay,
I
sir.
worked with starch-based plastics.
Okay, sir.
Okay, sir. If you can drop a question, email, we can discuss that, sir.
Okay,
Actually,
sir.
because this is not the right audience. Like, and then I cannot give a 1-minute answer to this.
Yeah. Okay, sir. Okay, sir. Okay.
Okay, sir. So we
Okay,
have
sir.
to see what is the extruder operating, what extruder you have, what are the zones in the extruder, how many zones, what are the temperature in each zone. All those things we have to see, sir.
Yeah, sure, sir.
Okay.
Sure, sir.
Sir,
Okay, sir.
I'll be happy to discuss with you.
Yeah, sure, sir. Sure,
Yes,
sir.
sir.
Thank you, sir.
Thank you, sir.
Sir, thank you for your lecture, sir. But I have one question, sir. You told about early pest detection and monitoring, sir.
Yeah.
So what kind of mechanism, like how is it applied in?
I have not covered something like nano sensors here, but I have given something in the write-up because I have limitation. See, one of the methods I arrange my presentation is our own experiments rather than telling generally our own experiments so that you can also get an idea what type of experiments you can apply like similar to. That is what my idea. So I have not covered nano sensors. Okay. So the quantity which is required is today, say somebody just wipes the towel, that is enough to identify the person because the DNA present in that can be extracted. Okay. So and then that can be like decoded, and then you can find out like so that level, like so the quantity of material which you require is very, very less in a nano sensor. Okay. So if we employ nano sensor, really you can detect any problem very early. Okay, even like people are telling now cancers and all, like they are telling they can detect 3 years ago, before 3 years before the actual symptoms are manifested. So they can find out because of the nano approach. Okay, so the early detection always helps because a stitch in time saves 9. Okay, so earlier the better.
My question is because like can this be applied to in relation to soil? Because with regards to food, the for example, if you're going for contamination or something, with regards to food, the contamination will be a little bit low, and the bacteria or whatever the contaminant is, the diversity will also be comparatively less. But when we're going for soil or pest detection in soil, it will be quite different because the diversity will be huge. So can it be applied or
Still, yes, you have to identify the compound which you are using as a marker for the pest. Okay, so you have a sensor, you use some principle for identification, okay, or measuring it. So for example, the insect releases a particular compound and the compound contains amines, let us say. So you develop a nanosensor for amines, which suppose other insects are by incidentally not producing it. So you have to identify the compound which is uniquely produced by, for example, in your case, the particular pest of your interest. Then develop a sensor for that. You have a ligand which binds with that, and then because that is where I was coming to that 6 micrometer this thing. You have one carbon atom in nanoscale. You have a billion. Okay, so you have a billion sites for binding as compared to one in a micro scale. So that is where very small early stage detection will be possible. Okay, yeah. So the principle is same. You whether you measure in food or in soil, you have to identify the compound which we are going to measure it. So you develop a sensor for that. Yeah.
Thank you, everyone.
Thank you.
Thank you, sir, for your valuable insights. Now I request Dr. Raviraj, Dean Engineering, to share a few words.
Silence, please. Silence. So, good evening to all of you. I can see a packed house here. So, I was just feeling that for the past 5 minutes, I should have attended this lecture. So many questions, so many queries have come because of the importance of the topic it has been taken today. And I should thank our Vice Chancellor and the Dean SPGS for arranging this very good endowment lecture on nanotechnology and food processing. And I should also thank our guest speaker here. He's alumni of our institute, and he has come all along from Bangalore to deliver this lecture. Not today alone. He came yesterday morning, and he is delivering to all the students. That is the dedication he has for the students, and it is not the end of the communication you have with the scientists here. Even if you respond, he told his mail ID, right? What is his mail ID? Yeah, you can respond to him, ask some good questions, clarification, keep in touch with him if you want to continue the professional development. He will respond to you definitely, and we will also invite him if you need him. We'll invite him again. Thank you so much, sir, for giving this wonderful lecture. And I'm sure that all the students are benefited. Even a lot of online people have come across. And I also thank our head of the department, food process engineering, for arranging this one at a very short notice. And I should also thank all of you for attending this one. I'm sure that all of you are doing the research or coursework research put together, right? Okay. Thank you so much for the opportunity.